So, photosynthesis. Good times, right? How many of you guys hate plants? Sweet. So, I once talked to somebody who was serious and they claimed to be a vegetarian. Not because they loved animals, but because they hated plants. And they wanted to kill as many as they could, or something like that. I don't know. So, I know. Photosynthesis. Good times. My laser is dying, and I'm sorry for that. Photosynthesis. Good times. You're going to see some really big similarities to electron transport chain. So, in fact, it is an electron transport chain of sorts. It's not the same one we saw earlier, but there are some glaring similarities. So, in this case, let's do a little compare and contrast. What was the final electron, the final guy to receive the electron acceptor in the electron transport chain? Oxygen, right? So in this case, what's the first thing? So that gets this kind of, and I shouldn't say it's really the first thing because it gets going a little bit, but what's the source of electrons for photosynthesis? Water. And what does it turn into? Oxygen. So it's starting kind of everything backwards. So the final electron carrier was oxygen in the electron transport chain. So in this case, it's going to be the first thing made instead. So instead of being used up, it's getting made. And instead of the last thing, it's the first thing. So and in this case, it's supplying the electrons, the first electron donor, if you will, not the final electron acceptor. But keeping that in mind, we have a bunch of complexes again. We're not calling them complex 1, 2, 3, and 4 anymore. So, but you're going to see some glaring similarities to the electron transport chain we saw earlier. So in this case, the point of the electron transport chain is very similar to what it was earlier. What was the point of the electron transport chain, the initial result? Proton gradient. That's the point here as well. So for photosynthesis, our goal is to create a proton gradient. Now, one thing to note, things are a little bit different now. So where did we try and pump protons in the electron transport chain? From where to where? Yeah, we're trying to pump them out of the matrix and into the intermembrane space, right? So notice the matrix was the very center inside compartment of the mitochondria, and we're pumping it out of there. So now we're doing the opposite. We're actually going to pump protons into the very center compartment of a chloroplast. So not mitochondria anymore, chloroplast. Chloroplasts also have an extra membrane. Maybe we should actually draw something for a change, since I don't have a slide on it. Correct. So there are three membranes in the chloroplast. There's the outer membrane, there is the inner membrane, and then you have these lovely things. So these are called thylakoids, and they're membrane-bound sacs. So they're called thylakoids, and when you put them in conjunction, they're called granum, or grana in plural. So these little membrane-bound sacs, that is the third membrane, and it's inside there that we're trying to pump protons. So now instead of trying to pump them out of the inside membrane, in chloroplast we're trying to pump them in. That is the goal of the electron transport chain. Let's fill that thylakoid space in there full of protons. That way those protons can do what? They can come back out and go right through ATP synthase and make ATP. So if we look at the division here, so we got the stroma here. So between the inner membrane and the thylakoid space, that's the stroma. So, and then inside this thylakoid space, or inside these thylakoids, that is the thylakoid space, and that's where we're pumping protons with the photosystems here. As they leak back out to the stroma, and I say leak, but as they come back through ATP synthase, they'll make ATP. All right. Take a closer look at photosynthesis. We usually split this up into two places, the light reactions and the dark reactions, right? So what's another name for the dark reactions? Calvin cycle. So the light reactions is the electron transport side of things we're going to talk about first. So, and that's to create that proton gradient. Why do we create a proton gradient? To create also ATP. It also is going to create one other thing for us. We're going to create not only some ATP from the ATP synthase, but the end acceptor of the electrons is actually NADP+, which gets reduced to NADPH. And that's helpful because NADPH is useful for anabolism, biosynthesis, and we're going to use it later on to make some stuff in carbon fixation. So two big points in this. The proton gradient is produced, which allows us to produce ATP, but also along the way we produce some NADPH. We're going to need both of those for the dark reactions later. So the whole point here, we need two things, NADPH, ATP, because we're going to use them up in the dark reactions. Now, the light reactions are called light reactions because they use light. They need light. Now, keep one thing in mind. We don't call them the light and dark reactions because the light reactions happen during the day and the dark ones happen at night. It's not the case. So these usually happen together, but only the light reactions require light. So we might have called the dark reactions the non-light reactions or something like that, but they call them the dark reactions. But it doesn't mean you have to be in the dark for those to be happening. They're happening all day long potentially as well, right alongside the light reactions. So it's just the light reactions are the ones that actually need light to take place. So we've got this big electron transport chain. So, and instead of starting at complex 1, so we're starting at photosystem 2. So notice it's kind of backwards. We're starting at photosystem 2. We'll hit photosystem 1 later. I tried to make this diagram look as similar to the other one. In fact, I used it as a template for making this one, just so we can relate the two as much as possible. So in this case, what is initially bringing in the electrons will be water, but actually that's not the first thing that usually happens. So usually we just assume this guy already has some electrons, and those get excited by light. We'll talk a little more about the chlorophylls involved and stuff like that. So, but they get excited, and that allows them to be transferred down the chain. So, and by transferring down the chain, photosystem 2 eventually hands them off to plastoquinone, which was remarkably similar to who? Ubiquinone or coenzyme Q that we saw. So, and it's soluble in the membrane, looks really similar, and plastoquinone then hands them off to cytochrome BF complex or B6F complex. Sometimes you'll hear it both ways. So this would be analogous to what in the electron transport chain? Not 2, complex 3. Complex 3. So analogous to complex 3 here. So, and then the cytochrome BF complex hands them off to a water-soluble carrier. So in this case, plastocyanin. And that's analogous to what? Cytochrome C that we saw in the electron transport chain. So directly analogous. So he then diffuses over not to complex 4, but to photosystem 1. So in photosystem 1 also requires the absorption of light. It promotes the electron to an excited state, allowing the electron to get transferred down the chain to ferredoxin. The ferredoxin dissolves away, diffuses away, takes those electrons over to FNR. This FNR contains FAD as part of its structure. So it's definitely involved in redox. And in this case, it is involved in then finally giving those electrons to NADP+, reducing it to NADPH. So the FNR here stands for FAD, NADP+ reductase. It contains FAD. It's reducing NADP+, hence the acronym FNR. And that's the final electron acceptor there, forming NADPH. So in the meantime, so where was the only place protons were actually pumped across the membrane? Yeah, the cytochrome B6F complex. So in this case, so in going from plastoquinone, which actually involves hydrogens, so as part of the oxidation reduction process, to over to cytochromes, which are iron, right? So, and don't involve hydrogens. Those hydrogens are now allowed to be given up and transferred across the membrane. So, but this is not the only source of the proton gradient here. So there's some here as well. If you notice, what's water converted into? Oxygen and H+. So that's another way of getting additional H+ on the lumen side of the thylakoid membrane. That's the thylakoid space side. So the other thing that happens is if you notice NADP+ turning into NADPH. I haven't written the complete balanced equation. Again, whenever I do like ATP and ADP reactions, I don't write inorganic phosphate. Same thing, I haven't been writing like NADH, so NADPH reactions, they also involve H+. And so in this case, NADP+ reacts with H+ when it gains the electrons to combine to form NADPH. So on this side of the membrane, we're actually using up H plus in the process. So on the lumen side, we create some H plus. So on the stroma side, we use up some H plus, and both of those also contribute to forming this proton gradient. Cool. So you want to remember the general outline here. We start at photosystem 2, go over to plastoquinone, over to cytochrome B6F, up to plastocyanin, over to photosystem 1, finally hand it off to what we call a soluble ferredoxin, which hands it off to FNR, and that's where NADPH is formed. Where's the only place, again, that protons are pumped across? So cytochrome B6F complex, and how many? Two. You want to know that. But again, is that the only source of the proton gradient? No. Also producing some when water is consumed and when NADPH is produced. So both those also lead to the proton gradient being what it is. And which side's going to have the higher proton concentration? The lumen side. And again, is that the inside or the outside of the thylakoids? Inside. It's the inside. So we're going to have higher proton concentration inside. That's, again, opposite of what we saw with the mitochondria. Okay, here's your lovely, typical Z-scheme. And how fun is this? Cool, your light reactions all take place inside that thylakoid space. They're on the inside part of the membrane, so in that thylakoid space, and that's where the reactions are happening. So we'll find out later that the dark reactions will actually take place out here in the stroma. So the Z-scheme kind of shows in an energy context. These are really common, things we show with photosynthesis. So one thing to note, photosynthesis is still kind of a gray area. It's still a place where active research is taking place. We don't have all the answers. We're kind of putting some stuff up here that we do know, and then we're also putting a few things that we think we know in here as well. But it's a little bit gray. So if you, stay at ASU for 10 years, you might see a little bit different diagram than the one you saw 10 years ago when you took biochemistry, or something like that. So just in case you happen to. I'd recommend it. It's a great place to be. Sweet. By the way, incidentally, I've been at ASU not 10 years. How long have I been there? 18 years. Isn't that fun? So which means I've been there half my life now at the end of this semester. Isn't that fun? It's just kind of a fun fact to say. I've been there half my life. All right, so Z-scheme here kind of shows you all the little bits and pieces. If you notice, we did this for mitochondria as well with electron transport chain. We showed how electrons went from complex 1 to complex 3 to complex 4. We saw how coenzyme Q allowed us to get from complex 1 to complex 3, and cytochrome C allowed us to get from 3 to 4. But then we went back through and said what's going on inside all the complexes. This diagram kind of takes all of that lovely stuff into account. Good times. So first thing, so what usually happens is P680. This is a chlorophyll. So chlorophylls somewhat heme-like looking in nature, but they're not heme. They're not iron centers. But somewhat heme-looking structures. And they absorb light. They're great chromophores, we say. So chlorophyll, why do they call it this one P680? Yeah, the max wavelength it absorbs. Over in photosystem 1, that's P700, because that's the max wavelength it absorbs. So these are two different chlorophyll involved. So, and they're just slightly different. They're very similar. So, and bacteria have very similar ones as well. We call them bacteria chlorophyll. And they just, by being slightly different, expand the wavelengths that might actually get absorbed. So they absorb not only in this red region of the spectrum, but it turns out they'll have some small absorptions in the blue region of the spectrum as well. So we also have some accessory pigments, carotenoids and stuff like this, that also will allow us to expand the different wavelengths we might absorb as well. So the idea is this. So at photosystem 2, there is one special chlorophyll molecule. He's the most important thing in the whole thing. And the goal is to get him some energy. But surrounding him might be hundreds of other less important chlorophylls. We call them the antenna chlorophyll molecules. And so you don't just have this one chlorophyll who's hoping to, for light to hit him. You have him surrounded by entire, I like to think of it like a big satellite dish, if you will, of chlorophyll. And they're all getting light. And then when they absorb light, they pass that energy all towards the center, to that one special chlorophyll molecule at the reaction center. So, and this is what we're talking about, is that one special chlorophyll at the reaction center. In photosystem 2, it's P680. You'll definitely want to know that P680 is associated with photosystem 2, whereas P700 is associated with photosystem 1. Cool. In receiving light of just the right energy, it excites that electron up. And so here we see P680 in the excited state. I think this thing, no, it's still there. I just can't find it myself either now. So P680 is in the excited state. So, and here's the deal. Without the light, you couldn't pass the electrons off. You can pass electrons off typically either in somewhat of a even fashion or downhill energy-wise, if you will. And so in this case, to pass these electrons off to some quinones here, and then off to eventually plastoquinone, P680 can't do it. He needs to get to his much higher energy excited state first. This is what the light's accomplishing for us. It is raising the energy of the electrons, so for P680, so that he actually can pass them off in a downhill fashion, in a spontaneous fashion. So they will eventually get passed off, and through plastoquinone, which is not on the diagram here, get passed off to cytochrome B6F. So kind of passes them energetically in a sideways fashion off to plastocyanin. They get over to P700. P700 would really like to pass them off up here and eventually pass them off to a soluble ferredoxin, but P700 can't until the electrons that he's waiting to pass off also get excited by just the right energy light. And once they're excited, they're much higher in energy, and now they can get passed down the chain. So, and eventually, again, they end up at FNR, which allows us to reduce NADP plus to NADPH. This is now the nitty-gritty. I honestly don't know how much you need to know of this. It seems like there was a little less emphasis of all the little tiny steps in the pathway than there was in the mitochondrial electron transport chain, but it is on one of the slides. So I would definitely, if you take a step back, you definitely, not step forward, you definitely want to know everything on this slide, every little carrier along the way. So going here, probably not the worst thing in the world. To cover all your bases, probably not the worst thing in the world to know every step along the way. Sorry.